You're watching a genomics education module produced by the Division of Genomic Medicine at UC Davis. This module will focus on different tests used for cytogenetic analysis, how they're different, and when to use each one. Let's start by talking about the different tests that are available for cytogenetic testing, or chromosome analysis. This module will walk you through the basics of karyotype, fluorescence in situ hybridization, or FISH, and microarray. These three tests are listed in order from least to most sensitive. We'll begin with karyotyping, or G-banding. This analysis will show you a snapshot of all the chromosomes in one of your patient's somatic cells. The chromosomes are depicted in a standard format called a karyogram, as shown here. They're organized in pairs, ordered by size from largest to smallest. A karyogram is helpful in determining if there are too many or too few chromosomes, or if the structure of any chromosome is aberrant, including issues such as insertion, deletion, duplication, translocation, or inversion. Karyotyping has a fast turnaround time. However, the resolution of the test is relatively low. In other words, an abnormality has to be structurally large in order to be identified by this test. Here is a list of a few common clinical indications for karyotype testing: recurrent miscarriages, male infertility, ambiguous genitalia, dysmorphic facial features or body structures, and developmental delay with anomalies. A more sensitive test is fluorescence in situ hybridization, or FISH. In this analysis, color tags adhere to specific regions of each chromosome. This indicates the presence or absence of certain DNA regions. FISH can also be used to track specific RNA targets in cells, such as tumor cells or tissue samples, which is useful for determining spatial temporal patterns of gene expression. One significant limitation of FISH is that it only analyzes the parts of the chromosomes where the probes hybridize. However, the probed areas are analyzed with high resolution. Here is a list of a few common clinical indications for FISH: suspected syndromic deletions, suspected trisomies and sex chromosome anomalies, or developmental disabilities with unknown etiology. Chromosomal microarray analysis is even more sensitive than either karyotyping or FISH. In this analysis, a patient's DNA is compared against a reference set of normal DNA. The different colored lights indicate where the patient DNA and reference DNA are the same and where they differ. The DNA is arranged in a specific pattern, or array, on a glass slide, so that any deviations from the reference DNA can be matched to the corresponding locus, or place in the genome. Targeted mutation analysis can then be performed on specific candidate loci. The major advantage of microarray is that it provides high-resolution whole genome analysis. However, it has a longer turnaround time and is relatively expensive. Here is a list of a few common clinical indications for microarray analysis: multiple congenital anomalies, suspected autism or developmental delay, patient carrying a fetus with one or more major structural anomalies identified on ultrasound exam, or suspected copy number variant or insertion deletion abnormality. Here is a table that outlines the major advantages and disadvantages of the three test types. So to recap, there are three main test types on the market for cytogenetic analysis: standard karyotype, FISH, and microarray. If you would ever like help deciding which genetic test to order, the Division of Genomic Medicine is here to help. Our clinic is located in the Mind Institute on the medical campus in Sacramento. You can refer patients to our genomic medicine clinic, visit our website, or call us with questions or to speak with a genetic counselor.